everybody, welcome to our video today where we're talking about biochemistry chapter 8, biological membranes. So before we get started with the contents of chapter 8, I just wanted to look at lipids quickly a little bit because I think there's some things that I did not mention in our lipid video, which was chapter 5, that are kind of important when we're talking about biological membranes. So we're just going to do a quick review of that before we get into chapter 8. So the biggest part of the lipids that I wanted to revisit was that there are 4 types of lipids in our bodies. So we have triglycerides, which is a glycerol molecule attached to 3 fatty acids via dehydration. And the reverse of this is the reverse of forming a triglyceride is a saponification. And a saponification is a base-mediated ester hydrolysis. So even that point right there is kind of important to remember and note, so definitely take note of that. Then we have fatty acid oxidation. So fatty acid oxidation, again, a triglyceride has 3 fatty acids in it. So fatty acid oxidation produces acetyl-CoA, and fatty acid synthesis fatty acid synthesis is you're using acetyl-CoA. So those are just important things about triglycerides and fatty acids. The second is a phospholipid, which is the main type that we're going to discuss in this biological membrane chapter. And that is a glycerol molecule with a phosphate group and 2 fatty acids. And you know, we have definitely spoken about this before, in having a polar head and hydrophobic tails. So then we have number 3, which is terpenes, and those are built from isoprene units, and terpenes are a precursor to cholesterol. And then we have cholesterol, which is the 4th type, and that's composed of 3 6-carbon rings and 1 5-carbon ring, and it's a precursor for steroids. Okay, so now let's move on to chapter 8. So the components of a biological membrane. We have phospholipids, which is, as mentioned, the primary lipid layer. We have cholesterol, which can increase the fluidity and stabilize the membrane. Then we have proteins, which allow transport. So we have different transport proteins and channels and things like that, which allow transport. And then we have carbohydrates, which are unique cell surface markers. So it just plays a role in the detection of certain cells. So kind of it's like it says a surface marker for certain cells. Okay, so next we're looking at just a couple of definitions in relation to biological membranes. So we have concentration gradient, which determines if it's active or passive transport. So the concentration gradient is built when you have a higher concentration on one side of the membrane and a lower concentration on the other. Of a solute or a molecule or anything like that. And so we'll get into it in a moment, but active transport is when you're bringing molecules against their concentration gradient, and then passive transport is when a solute is going along its concentration gradient, and going along its concentration gradient means it's going from a high to low concentration. So it does that so it can try to reach equilibrium. So again, the concentration gradient will determine the type of transport, active or passive. And then we have osmotic pressure, which is the pressure applied by a solute to a solvent. Okay, pretty an easy one to remember. So if we have a glass of water and has a lot of solute in it versus a glass of water that only has a little bit of solute in it, the one with more solutes in it will have a higher osmotic pressure because there's more pressure being applied to the solvent because there's more solutes exerting that pressure, if that makes sense. Then we have ligands, which are extracellular ligands combined to membrane receptors and cause secondary messenger cascade pathways, which we talked about in a couple of chapters as well. Then we have gap junctions, which allow for rapid exchange of ions or small molecules between adjacent cells. And then we have tight junctions, which prevent solutes from leaking into space. We have flipases, which maintain a bidirectional transport of lipids in phospholipid bilayers. I hope you can see this down there. Okay, and then finally at the very bottom it just says lipid rafts, and it says freely moving lipids in a phospholipid bilayer. Okay, so now that we got a lot of those definitions out of the way, let's move over here where we're talking about active transport, passive transport, and then just two other definitions. So membrane potential is maintained by the potassium sodium pump and lake channels, and the resting membrane potential is at approximately -70 millivolts. Now this whole process is explained in chapter 4 when it relates to the nervous system. We look at how action potentials and that membrane is, and that resting membrane potential is kind of maintained. So definitely look at chapter 4 video for that nervous system. But just to note there, because we are talking about the membrane, so that is membrane potential is maintained by the sodium potassium pump and leak channels, and resting membrane potential is approximately -70 millivolts. So we have endocytosis and exocytosis, which is the method of taking in, for endocytosis, or releasing, exocytosis, substance into a cell or out of a cell. So pinocytosis is the ingestion of liquids via vesicles, and phagocytosis is the ingestion of larger solid material. Okay, so now we are at active transport. So for active transport, as mentioned, you are moving solutes against its concentration gradient. So if it's going against the concentration gradient, that means you're bringing it from low to high concentration. And because that is not just naturally what it would want to do, because naturally it would want to go from high to low concentration, right? You're bringing it against the gradient. You're bringing it from low to high concentration, which means you're going to require energy, in this case ATP, to do that, to move it against the concentration gradient. And so you have primary active transport and secondary active transport. So primary active transport directly uses ATP to transport molecules. And secondary active transport, uses coupled transport to harness energy released by one particle going down its concentration gradient, and the energy that is harnessed by that is used to power another. Another two important things about this, you have symport or antiport. So symport is during the secondary coupled transportation. Symport is when both particles are flowing in the same direction. So you have this molecule here that wants to go along its concentration gradient, and as it does that, the energy harnessed by that is also bringing another particle in the same direction. But the second particle here requires the energy produced by the first solute going through, if that makes sense. But again, just to reiterate, secondary transportation harnesses the energy released by one particle going down its concentration gradient to power another. And so symport is when both of those particles are going in the same direction, and antiport is when those particles are flowing in opposite directions. So one's particle is coming in, the other one is going out. Then we have passive transport, which is when no energy is required to move the solute, because it's moving along its concentration gradient from high to low concentration to attempting to reach equilibrium. So you have simple diffusion, which is for small non-polar molecules that again go from high to low concentration. Then we have facilitated diffusion, which uses transport proteins to move solutes across. So even if it is going with its concentration gradient from high to low concentration, sometimes additional protein channels or just protein transporters need to be used depending on the polarity of the molecule or the size. It might not be able necessarily to just go through the membrane. It might need a specific channel to go through. Even if it is able to go through with its concentration gradient, it might not physically be able to go through, even if it wants to, which is why you need some protein transporters for that. And so that is called facilitated diffusion, because that transport needs to be facilitated by a transport protein. And then finally we have osmosis, which is the movement of H2O across a membrane from high to low concentration. So again, kind of all using the same ideas, but different types of transport. But keep in mind the the concentration gradient. That's kind of the biggest takeaway from this chapter, is the composition of the lipid bilayer and looking at what molecules are trying to get through this membrane, polarity, size, things like that, and what does the concentration look like on either of these sides, so you can determine, if it's active or passive transport, and if you need ATP or things like that. So thank you for listening. If you have any questions, please put them below, and if not, we'll see you in the next video.